Now I will move on to our next presenter, Presenter Das.
Thank you, Christine. And
Yes, we can hear
so,
you. Thank you.
okay. So, good afternoon, everyone. And I'd like to begin by thanking the organizers of this excellent workshop. I have really learned a lot in these 2 days. I am Lopamudra Das, and I am working with Dr. Christopher Wohl at the Lunar Dust team at NASA Langley. And today, the topic for my presentation is the Lunar Simulant Deposition for Evaluating Adhesion. Now, this talk was previously given by Dr. Wohl at the Dust Mitigation Technology Developers Working Group. And so some of you might have seen this before. And so just to say, we have not had a chance to implement any of the excellent suggestions that we had from that talk, but we are working on it. Next slide, please. So as we know very well during this conference that lunar dust is a huge challenge. We see on the left-hand side the image of an astronaut. His glove is covered with lunar dust. And on the right-hand side, we see a vision of the future where we will have long-term lunar bases, and hopefully we will have conquered the problem of lunar dust adhesion. And so we are trying in our group to develop passive dust mitigation materials. And so one part of this is finding the right materials that will work in the extreme lunar environment and develop surfaces that will mitigate the dust adhesion. And so for this particular experiment, we have two parts to the study. So one is we have to develop in a lab a way to contaminate the surfaces with a fine monolayer of dust particles. And the second is we will have an adhesion testing system to see how well the materials perform with respect to the adhesion test. And at each step, we have optical microscopy to characterize for how the samples are performing. So the lunar environment is very different from the environment on the Earth, and we cannot have all the conditions simulated, but we simulate some of the conditions. And for our dust, we have used the LHS-1D, which has a particle size of less than 30 microns from Exolith Lab. Next slide, please. So before I go on to how we contaminate the surfaces, I'll give a brief overview of how we are going to characterize the adhesion force. So we have basically an adhesion testing system which consists of a sonic wand and an optical particle counter. So the sonic wand is vibrating at a fixed frequency, but its amplitude can be varied. And then, so we attach the dusted sample at the tip of the sonic wand with an adhesive, and then we change the amplitude according to a fixed recipe. So the particles that are there on the surface of the sonic wand, when the amplitude gets increased, they are detaching and falling into the optical particle counter in the funnel there, and they're detected by the particle counter. So one step of the process is we can detect the number of particles by the optical particle counter. And then at each step, we also take optical microscopy images to see how well the test is, the materials are performing in the adhesion test. Next slide, please. And so this is a very simplified view of how things are happening at the tip of the instrument. So we have the adhesion force of the particle on the surface. We have the force of acceleration. And of course, there is the force of gravity, but because the particle size is so small, that is very negligible. So the particle will basically detach when the force of acceleration is greater than the force of adhesion. And when they detach, they are detected by the optical particle counter. Now, as I've said before, this is a very simplified version, and the actual interaction is way more complicated than this. And next slide, please. So what we are trying to do is as we can see on the left-hand side, we are trying to deposit a monolayer of particles. And so the adhesion forces that are mainly working here are van der Waals forces, electrostatic forces, magnetic, chemical, and mechanical forces. And on the Earth, we also have the force of gravity and capillary, which is very important in the ambient atmosphere. So in the actual case, we are trying to deposit particles. We are seeing that cohesion force plays a huge role, and there is also mechanical interlocking and it requires quite a bit of effort to get a monolayer, because in most cases we are seeing an aggregate of particles. Next slide, please. So this is a schematic of how we tried two different methods. One was the aerosolization method, and one is the vibratory filtration method, to deposit a uniform monolayer of dust particles on our surface of interest. So in the aerosolization scheme, we have an enclosure, and in the enclosure, we have a Hirsch funnel which has perforations in it. And so the Hirsch funnel is attached to a compressed air canister, and we place the sample with a mask on it inside the enclosure. And then we put a fixed amount of simulant into the Hirsch funnel, and then we take a couple of bursts of air through the compressed air canister and basically aerosolize the dust and allow it to settle on the surface of the sample. And then after everything is settled down, after a couple of minutes, we remove the sample and take off the mask to have a uniform area, that window that was exposed to the dust, and then we see how that went. In the vibratory filtration method, we have a sieve system. And so the sieve basically has a vibratory motion and a tapping motion. And so the sample is placed underneath the sieve, and we vary the time, we vary the number of taps and the amplitude of the vibration to see how the deposition occurred. Next slide, please. So this is how the system looks like. We have the funnel and we weigh the simulant to be dispersed in the aerosolization chamber. So we varied the amount of simulant from 50 milligrams to 250 milligrams. And so the samples you can see are placed at the back, and we have actually changed the setup since then, but the basic concept remains the same. Next slide, please.
Can we get the audience to mute your mic? We have a hot mic.
Okay. Do I continue?
Yes, yes, please continue.
Okay, and so this is how the simulant deposition to the vibratory filtration looks like. We have this Gilson sieve shaker, and we have two sieves. So sieve number one has 32 micron opening, and sieve number two, so the one on the top is the one with the higher opening, and then at the bottom it's a 50 micron opening. So basically we put the simulant on the top sieve, and then we set the amplitude at a certain amplitude, and then also we put the number of taps. So each tap motion takes 1 second. So by increasing the number of taps, basically we are increasing the time that the deposition is occurring. And we varied the simulant from zero point 0.2 to 2 grams. Next slide, please. So this is how, so we did a lot of samples, and this is just a very brief sampling of what we found. So this is in the aerosolization technique. As we can see, in sample number 39 on the left-hand side, we had 250 milligrams of sample simulant that we used to aerosolize. And that actually resulted in a lot of clustering of the particles on the surface. And next to it, in sample number 42, we had only 50 milligrams, 1/5 of the earlier amount on the surface. And here we see that there is very little dust particles that we can actually see. So that was one. And then in the vibratory filtration method on the right hand side, simulant mass in both cases was taken to be 1 gram. But in sample number 10, the number of taps was 20. The amplitude was 5. So 20 taps means 1 tap per second. So it was a 20-second experiment. But in sample number 35, it was amplitude was 4. So it was very close, but the number of taps was 300. So it ran for an entire 5 minutes. And we see that the position is relatively uniform and even. Next slide, please. So then because the discrepancy between the 50 and the 250 was so much, we decided to do some more variations in between. So here we are showing on the left hand side in the aerosolization method, we had the simulant mass at 150 milligram and the substrate one was glass on the left hand side, and on the right hand side it's titanium. And we see that there was a reasonable amount of dust coverage on the sample surface. In the vibratory filtration method on the right hand side, we see in sample number 44, we did what we did previously. We had the same 5-minute run, number of taps was 300, and the amplitude was 4, and the simulant mass was 1 gram. However, this sample looks very different from what we found in sample number 35. And then sample number 36, the same experiment had happened, and we found that there were areas where there was no dust coverage. So what we found in the vibratory filtration technique, that there was no reproducibility from sample to sample, even if all the parameters were kept the same. Even though in some cases we were getting really uniform and monolayer-like coverage. Next slide, please. So we decided to do something else to see if we could get a reference for all samples, like a baseline. So what we decided was, after we deposited the dust, either by aerosolization method or by vibratory filtration method, we would run the samples at a very low setting of the sonic wand, just to remove all the cohesively attached particles. So this would be at the lowest setting of the sonic wand and for a very small amount of time, which is fixed for all the samples. And so what we are seeing is that on the left hand side, after aerosolization, the simulant mass was 250 milligrams and it was thick dense clusters. After sonication, all the thick clusters went off, they were detached. The larger particles were also detached, and it's only the smaller fine particles that are evenly distributed on the surface. The same thing happened in case of the vibratory filtration method. So after the low sonication method, we find that only the smaller particles remain and all the larger cohesively attached particles have detached. So after this first step, basically, the aerosolized sample as well as the vibratory filtration sample, they both have kind of a uniform distribution of particles. Next slide, please. So then we decided that, okay, the aerosolization technique is a more efficient one because the reproducibility is more. And so we decided to go for the aerosolization technique. And as we can see, this was one of the results of the experiment. We varied the simulant from 50 milligram to 250 milligram. And we found out that the sweet spot was at 150 milligram, where we were getting less number of clusters and a reasonably even distribution of the particles. Next slide, please. So this is how we came to determine the ideal approach to the surface contamination for our adhesion study. The first step is we prepare the samples, the materials, the surfaces, and then we take a small section of the sample, we place it with adhesive on the sonic wand tip, and then we have a mask to expose a fixed window on the surface to the dust. Then we place the entire sample holder into the enclosure of the aerosolization chamber, and then we put in 2 short bursts of compressed air through the simulant in the harsh funnel and allow it to aerosolize, settle on the sample, and wait a few minutes. Then we take out the sample container, take off the mask very carefully, and we have an even distribution of the dust in that exposed window. And
2 minutes.
afterwards, yes, thank you. And then we resolve the cohesion by putting it through a low displacement sonication method, like for at the lowest setting of the sonic wand for a very short amount of time, which is fixed for all the samples. And after that, we have a uniform distribution of particles on the surface. Next slide, please. And so here is the rest of the Langley Lunar Dust Brigade. We have Valerie Weisner, Keith Gordon, Glenn King, Sang Choi, Jonathan Hernandez, and Catherine Crossett. And we have researchers at nearly every NASA center and collaborators throughout academia and private industry. And unfortunately, we could not get a group photo. We were hoping to get one. And in the next slide, please. I'd like to acknowledge Dr. John Connell of the Advanced Materials and Processing Branch at NASA Langley Research Center. From the NASA Lunar Surface Innovation Initiative, Nikki Werkheiser, Michael Johansson, Erika Montpack, Kristen John, and Cameron Hartman, and also NASA Internship, Fellowship, Scholarship, and STEM Text Flight programs. Thank you, and I'm open to any questions.
Okay, I think we have time for one question from the chat. Do you control moisture in any way?
So far, we have put in a desiccant in the dust chamber, but since the air contains a lot of moisture, I would say that that's not sufficient. So we are looking to find ways to mitigate the moisture even further.
Okay, thank you.